Okay, first some background. The experiment is going to involve bacteria. Now, this is a scanning electron microscope picture of some bacteria, and they've been colorized red here. So these are single-celled organisms. They're the smallest cells on Earth. Very simple. They do not have a nucleus, but they have a membrane and sometimes a cell wall, and they have DNA in them and cytoplasm and so on, all the basic characteristics of the cell. In fact, in some of these individuals, you can see that they are dividing. So bacteria reproduce by cell division. One cell is splitting into two. Now, if you take these bacteria and you put them in a suitable environment, they will grow. They will take in nutrients and reproduce and increase their numbers. And so here we see a picture of a petri dish, and on the surface of the petri dish inside there is a food medium of some kind, and each of these little spots here is a colony of bacteria. A colony of bacteria would be maybe 1,000,000 individuals. Each of those 1,000,000 individuals descended from the original founding bacteria, like this one right here. Maybe that one is this gave rise to this colony right here. Now, to understand this experiment, it's necessary to know that bacteria, while often thought of as possibly dangerous to us, as when we get a bacterial infection, they themselves can be the victims of a viral attack. In this electron microscope picture, the bacteria is seen here. That's the surface of the bacteria. And these strange objects that are attached to the bacteria are viruses. They're called bacteriophages. And each of them has anchored itself to the surface of the bacteria, and it will inject its genetic material into the bacteria. That genetic material has the information to make viral parts. And so what the virus does is it uses the bacteria cell to reproduce more virus particles. It hijacks the cell. It is a parasite. And when these viruses burst out of the bacteria cell, here you can see the bacteria has been destroyed here, it kills the bacteria. These new virus particles can go on to infect other bacteria cells. Now, the fact that viruses can kill bacteria cells is going to be useful for the plan of this experiment. Because what the researchers are going to do is they're going to have some bacteria growing quietly, comfortably in a petri dish, and then they're going to add virus to the plate. So in this way, they're changing the environment of the bacteria. And what they wanted to know was whether the presence of the virus would somehow cause bacteria to produce variations in their offspring that would be resistant to the virus. In other words, could the virus cause the bacteria to start to have useful variations in their offspring? So it's not God that would be biasing the reproduction, but maybe the environment itself can bias the reproduction of organisms. So in this way, the environment would cause bacteria, the virus would cause bacteria to start producing offspring that were resistant to the virus as a way of sort of combating the new environmental change. So researchers set up an experiment to test this possibility. Okay, so here's the situation here, and I'll just sort of describe this one step at a time. Initially, what they did was they set up a petri dish, and they plated some bacteria on that dish. And so here's bacteria cell number 1, cell number 2, cell number 3, 4, and 5. That would be a single bacterium. Now, if there's enough food in the solution of the petri dish there, then they will start to divide and reproduce. And pretty soon you'll get colonies. And all of the 1,000,000 of individuals in colony number 1 can trace their ancestry back to bacteria number 1. So they would be clones. However, if during reproduction, if there's any change, if there's new variation introduced, then there will be some variation in individuals of that colony. But since there are only a few generations to make 1,000,000 individuals, the chances of there being too many new mutations would be small for this little colony. However, it is likely that colony number 1 is different from colony number 2 because bacteria number 1 and bacteria number 2 may have already some genetic differences. So we should expect that all the individuals in colony 1 are pretty much clones, identical individuals genetically, but that colonies 1, 2, 3, 4, and 5 might show some variation because bacteria cells 1, 2, 3, 4, and 5 might have been a little different to begin with. All right, so here we go. We've got a petri dish with 5 colonies of bacteria. Then what they do is they make a duplicate plate, and they do that by pressing a piece of felt onto the surface of this plate, and that will lift up a few individuals from each colony but preserve their spatial location. Then they take that felt and press it down onto a fresh new petri dish, and so you reestablish colonies of bacteria. So these 5 colonies should be clones of those 5 colonies. So we have a duplicate plate. Now, the next step. At this point, they add the virus. The very virus that can destroy bacteria by attaching to its surface and injecting its genetic material and hijacking the cell to produce more virus that kills the bacteria. So they add virus and they watch what happens. Sure enough, colonies 1, 3, 4, and 5 were destroyed by the virus. Dead colonies. But in this case, it happens that colony number 2 survived. So somehow, colony number 2, those individual bacteria cells, had some trait, and now we can call this an adaptation, because that trait allowed them to survive in the presence of the virus. That was an adaptive trait. So the question here, critically, was, did the virus itself cause colony number 2 to become resistant to the virus? Or was colony number 2 already resistant to the virus before they even added the virus? Those are the 2 possibilities. That's what's at issue here. Can the new environment cause useful adaptations to occur? Or is the environment simply selecting useful traits from populations that already had differences in traits? Well, to get at that question, what they did was they went back to the original plate and they added the same virus to the original plate. Remember, that's a copy then. The duplicate plate is a copy of the original plate. And when they add the virus to the original plate, sure enough, what they found was colonies 1, 3, 4, and 5 were destroyed. Colony 2 survived. Now, if you do this once, you might say, well, that could be a coincidence. It could be that the virus happened to change colony 2 to become resistant down here and happened to do it over here as well. But they repeated this process many times, and they found the same thing. If it was colony 2 that survived here on the duplicate plate, colony 2 survived on the original plate. If it was colony 1 that survived on the duplicate plate, it was colony 1 that survived on the original plate. If it was colony 5 that survived on the duplicate plate, it was colony 5 on the original plate that survived. In other words, it was always the same colony that survived. For those researchers, the most likely explanation was that the colony that survived here on the duplicate plate was already resistant to the virus. And so since it was already resistant to the virus, that same colony on the original plate would also be resistant to the virus. And so when you add virus, it's the same colony that will survive. Now, to really see the reasoning behind this, think about the alternate possibility that the virus would be causing a colony to become resistant. So let's go back to, let's take a look at these plates here. So here's the duplicate. Let's say this colony right here was resistant to the virus on the duplicate plate. Now, if the virus caused that colony to become resistant, then you might expect that on another day, when you do this experiment, the virus might have caused a different colony to become resistant. But on this particular day, it caused this colony right here to become resistant. So when we take the virus and put it back on the original plate, we might expect that the virus might turn some other colony into a resistant colony. But no, the virus seem to be unable to destroy the very same colony as was on the duplicate plate. Again, if the virus were causing this colony right here to become resistant, you might think that when you put it on the duplicate plate, the virus would have the power to cause some other colony to become resistant. But that's not what they found. It was always the same colony that was resistant. So again, the most likely explanation for these researchers was the virus was not causing a colony to become resistant. Rather, the virus was selecting from among variation on the plate, variation among these bacteria colonies. The virus selected those colonies that were resistant to the virus, destroying the other colonies. And the reason why it was always a matching colony on these two plates is because that variation was present at the beginning of the experiment, before the virus was even introduced. So this suggests that in nature there is all kinds of genetic variation in populations. And when a new change in the environment occurs, that change in the environment will be selecting individuals with the most useful variations. But the variations themselves already existed. They were not caused by the environment in such a way that the environment could cause useful variations. Rather, the variations were present, and we'll learn later that these variations are the result of genetic differences among individuals. So this experiment gave support to the idea that neither a divine influence or the environment is shaping the kinds of variation produced in a population. Biologists argue that these variations are randomly produced, creating individuals, many of which are like their parents, but some of which are better adapted to the environment and some of which are worse adapted to the environment. And it's the environment that selects individuals in this random population. The environment does not cause useful traits.